All right, ladies and gentlemen, welcome back. This is part 4 of our Biomolecules unit. We're going to be looking at carbohydrates and lipids, essentially just the structure and the function. So when we look at carbohydrates, the essential function of carbohydrates is energy, production, and use. And if you look at the empirical formula, which means the basic relationship between the number of each of the element's atoms, it's CH2O, which literally translates to H2O, hydrate, and C is carbon, so carbohydrate. So if you want to be able to recognize a carbohydrate, it's going to have a ratio of twice as many hydrogen atoms as it does carbon or oxygen. Okay. So monosaccharide is the first structure that we're going to be looking at. This is the monomer of carbohydrate, as we talked about in the last unit. So here are some examples: glucose, fructose, and galactose. All of their formulas are actually C6H12O6, which is twice as many hydrogens as carbon and oxygen. However, they're considered isomers, which means they have the same formula. The only differentiation is how they are actually arranged. And if you look at these examples, you can see it. So here is the glucose. Galactose is just a change in one of the OH bonds. Instead of being on the right side here, it's on the left. And if you look at the carbon where it's double bonding to the O, it's on this fifth carbon here instead of the sixth, like glucose and galactose. So just subtle little differences lead to a big difference in function. So you can jot those down. You have these pictures in your notes. Another example is also ribose and deoxyribose. These ones are used in DNA and RNA. So the disaccharide is essentially two saccharides, two carbohydrates. We consider them two monosaccharides, and the way that we join these together is through that process we talked about last day, which is dehydration. Okay, so here it is. We have glucose monomers, monosaccharides. On each side you have an OH and an H group. When they come together, the H and the OH come off and form water, and that leaves an attraction between these two glucose monomers, and it ends up creating this disaccharide called maltose. Okay, so you should have that picture as well. Another few disaccharides that we'll be using and talking about is sucrose, which is essentially when you have glucose and fructose coming together. The one we just talked about, two glucose molecules come together to form maltose. And then we have lactose, which you find in milk, and that is the sugar that's composed of glucose and galactose. Notice the endings, all OSE, an indication of a carbohydrate. So polysaccharide, the last part we're going to talk about with carbohydrates, it's when we have three or more monosaccharides. It's usually actually a longer chain. We lend the term oligosaccharide to anything between 3 and 9. But the big ones are the polysaccharides. These ones, again, are joined through dehydration. When you put that third monosaccharides on there, more water is given off and creates that bond. Here are the ones we're looking at. We have starch, which is energy storage in plants, cellulose, which is structural support in plants, and finally glycogen, which is energy storage in animals. So when our blood sugar gets too high, we got to get rid of that glucose in our blood. It will be stored as glycogen in our liver. So you can pause that to get it down. And we're going to look at the structure of them here in a second. So here's the structure of starch. So it's a polymer of glucose. Here's the structure of cellulose, polymer of glucose, and glycogen, which is a polymer of glucose. So they all have the root structure of glucose, but the way that they're bonded is differently. So if you look at starch, all of the glucose molecules are bonded with the oxygens all in one direction. And if you compare that to cellulose, it has oxygen up here, then down below, then up. Same with the CH2OHs, they're all on the top here. But if you look at cellulose, they're alternating from the top to the bottom. So that difference in structure leads a difference into their function. Instead of storage of energy, it's a structural difference. Okay. So the way I kind of remember that, starch starts with S, so does same. These are on the same side, so that's starch. You compare that to glycogen, it's very similar to starch in terms of its bonding, but the big difference is that the glycogen is highly branched. So there is some branching like, a tree in starch, but if you look at this one, it's not the best picture, but it shows that this one's branched. So that's the difference. So you're going to have to know them in terms of their function and their structure. Okay? So lipids, it is a little bit different function. It's also used for protection. You have the fats around your vital organs, so if you get trauma there, it hopefully won't cause too much damage. And lipids are also made up of carbon, hydrogen, and oxygen, just like the carbohydrates were, but it won't be in the same ratio. It does not have twice as many hydrogens as it does carbon and oxygen. So what they're made up of, we don't consider a lipid a polysaccharide. So when we talk about their subunits, we don't call, as a polymer, we don't call their subunits monomers, because essentially a monomer should be able to be multiple put together in order to make one polymer, but that's not the case here. So what they're made up of is a molecule called glycerol. It's got the 3 carbons, and it's got H's on each side and OH's on the other, just like the other monomers. It has a fatty acid chain as well. So when those fatty acids and the glycerol come together, that's what's going to make our lipid. Okay? So the process of creating a lipid is also dehydration, where we take 3 fatty acids and add it to the glycerol. There's 3 carbons in order for these fatty acids to join. So if we look here, what happens is the OH from one side of the carbon is going to be taken off, and as well as the H from the carboxyl group on the fatty acid. And when those come together, it gives off H2O. And when those are given off, it creates a certain charges that are going to be attracted and allow these to come together. So when we have 3 of them come together, what it creates is this thing called a triglyceride. We also mention it as a lipid or a neutral fat. So you need 3 fatty acids coming together with the 1 glycerol to make up your fat. Okay? So the other variation of a lipid is a phospholipid, and it's a major component of the cell membrane. Okay? It creates a phospholipid bilayer, which we'll see when we get into the cell. So it's like I said, it's a variation of a triglyceride, and one of the fatty acids is replaced by a phosphate and a nitrogen group. And what it does is it creates this polar region in this otherwise non-polar molecule. And so if we look here, so this was the triglyceride formerly, where we had that 3rd fatty acid, but instead we have the phosphate group and the nitrogen that branches off instead and gives us that polar charged part. Just as a little addition here, we don't tend to draw them like this. We tend to draw it with a circle like a phosphate head and then these 2 tails, and it kind of looks like this. So this is an example of our phospholipid bilayer. We have our polar heads and then our non-polar tails. These polar heads are hydrophilic, which means it likes water, and these guys here are hydrophobic, which means they're scared of water, so they tuck their tails in and it creates this cell layer. And then finally, steroids. It's a completely different structural arrangement. It's composed of 4 fused carbon rings, and it looks like this. They're not going to be mistaken because you don't see these types of structures very often. And some examples that you've probably heard of before are estrogen and testosterone. They're the sex hormones. We'll be looking at those in reproduction. And then cholesterol, which is also in the cell membrane, and it adds to the integrity of that membrane and prevents it from essentially becoming too fluid. Okay, so if you have questions, feel free to pop in and ask. Otherwise, we'll be touching on this in class.